This video was designed to be seen by clients before a nutritional blood microscopy. The first thing you'll need to do is prepare for your nutritional blood microscopy, and what you need to do is consume a meal that contains protein, carbohydrates, and fats about 4 hours before your appointment. During those 4 hours, just drink water. So proteins are usually found in meat, eggs, and beans, nuts, and seed. Carbohydrates, generally fruits, vegetables, and grains. And fats are usually with the protein foods like eggs and meat. Fats are also found in oils and, again, nuts and seeds. So it shouldn't be a problem to consume foods containing protein, carbs, and fat about 4 hours before your appointment. Then just drink water during those 4 hours. The reason you do this is because I want to see how well you're digesting proteins, carbohydrates, and fats. And if we wait 5, 6, 10, 12, 20 hours, perhaps you've digested that meal, but you really should be able to digest a meal in about 3 hours. So we can see evidence whether you're digesting these macronutrients, proteins, carbohydrates, and fats. When you arrive for your appointment, you'll be filling out a little form, very simple, name, date, address, and that's about it, a very brief form. And then you'll sit to my right, and I am going to take a drop of blood from your left middle fingertip. I use a lancet with a very thin pin or needle. I always tell people I won't feel a thing, but actually you probably won't feel anything either. I'll wipe away the first drop and I will utilize the second drop. I'll grab that second drop with a little cover slip, and it's going to be hard for you to see, but I'll take that cover slip and I will sandwich the blood between the cover slip and a slide. Because of that, there's going to be some room air that gets trapped with your blood under the cover slip and slide. It is not air in your blood, it's air from the room. And that's going to look purple using my high magnification lens, and my lower magnification lens, it'll look like a big circle with a white border. So when we see that, we know it's room air. So the first thing I'll do once I have your blood under the microscope lens is I will scan at fairly low magnification for crystals. So the crystals are typically plaque, uric acid, and cholesterol. Plaque is a mixture of LDL cholesterol mixed with fats, oils, your own blood platelets, and bacteria. Just like the plaque on your teeth is a mixture that hardens, well, the plaque in your blood is a mixture that hardens. The main component is usually LDL cholesterol, but there's also a big component, and that's calcium. So medical doctors call plaque calcification, but really it's this mixture of fats and oils and calcium and bacteria and LDL cholesterol. The other common crystal is known as uric acid. It will be a bright yellow crystal, looks like a piece of broken glass. And I always get this question, what is that? How is it formed? And how do I get rid of it? So I'm going to answer it right up front. Uric acid forms when there's some combination of incomplete protein digestion combined with not enough calcium intake, so not enough dietary calcium. So how do you solve it? Well, if you're not digesting proteins, you may need a plant enzyme. If you're not taking in enough calcium and not absorbing it, some of that is hereditary and some of that is genetic, but you may need to supplement with a plant-based calcium or just eat more calcium-rich foods. The calcium-rich foods are typically the green vegetables. You notice I didn't mention dairy. So dairy comes with some issues. It is one way to get calcium, but it is not the preferred way. The preferred way are the green vegetables. especially cucumber and asparagus are quite high, but the leafy greens, all good ways to stop forming uric acid crystals. During a follow-up analysis, if you can get rid of the uric acid crystals with just some dietary changes, great job. If not, you'll want to supplement with a plant-based calcium, in this case, coral calcium. Coral is plant-based. So after I'm looking for crystals, I'll also be zooming in with my high magnification lens, and as I see white blood cells, I'll stop and point them out. We want the white blood cells, which is your immune system, to be large, and I'll explain whether a particular white cell is large. And we also want the inside of the white blood cell to be moving. So we want your immune system to be moving, and we want the white cells to be large. So the size of the white cell is analogous to strength of your immune system, and that movement within the white blood cell is analogous to activity. So we want a strong and active immune system. The white blood cells are quite important, but the ratio of red blood cells to white blood cells is about 700 to 1. So when I find a white blood cell, I typically zoom in on it. Okay. The other thing you'll notice once I get to high magnification is you'll see a lot of movement, and these are the red blood cells moving. So you will think perhaps the red blood cells are alive and they're swimming, but that is not true. The red blood cells are not living cells. The white blood cells are not living cells. Your platelets, the 3 components of your blood, also not living cells. So humans, when we put them all together, they're alive, but most of the cells in the human body are just that. They are cells. They are not single-celled organisms. The reason the red blood cells will be moving is the whole drop that's on the slide will be flattening. And in this process of flattening, the red blood cells are along for the ride. So it really looks like they're moving under their own power, but they're not. The drop is just flattening. However, it's very good if the red blood cells are moving, because that means they're not clumped together by undigested proteins. So proteins, when they're undigested, tend to clump the red blood cells. Fats and sugars, when they're undigested, tend to clump the platelets. So if we have movement and individual red blood cells bouncing off each other, that's a good thing. Now, the main part of the blood analysis, I'll be filling out a form like this, and I will be looking for irregularities in the red blood cells. They should be round in shape, all about the same size, and no imperfections on the membrane. But we may see some imperfections, and I'll go into what that means. There is a living component in the sample, perhaps, and that would be yeast, bacteria, that would be parasitic bacteria, or fungal form. So if I see that, I will identify it and tell you what you need to do to get rid of it. Now that's the main part of the blood analysis. This is a nice introduction for those of you to watch a day or two before your blood microscopy. If you have questions, please leave them in the contents, and as always, thanks for watching.